Okay, welcome to the video reviewing the Krebs cycle, also known as the citric acid cycle. This is the second stage in aerobic respiration. Remember, anaerobic respiration does not involve this step. I recommend that you follow along with a piece of paper and you draw what I'm drawing so that you can gain a better understanding of what's going on. Krebs cycle takes place inside mitochondria. So I'm going to draw a large mitochondria in here. Take up most of the space on the page. Remember, mitochondria have a double membrane. The inner membrane is folded. I'm not going to exaggerate those folds very much at all, but I'm going to flatten them out a bit. In reality, these are much more pronounced folds, going right into the matrix. These folds are called cristae, remember. Okay, so let's talk about Krebs cycle and what happens in it. There's going to be a lot going on in this diagram, so make sure you leave plenty of space. First of all, we start off outside the mitochondria. Remember, the process glycolysis produces pyruvate, and it produces 2 pyruvate molecules for every 1 glucose molecule. Pyruvates are 3 carbon molecules, so we have 2 of those going into the Krebs cycle. Before they get there though, they take part in another reaction called the link reaction. And the link reaction is basically a way of transporting the pyruvate in and getting a little bit of energy out of it as we do so. In the link reaction, NAD plus is converted into NADH. 1 molecule of carbon dioxide is released, and that leaves the cell. We breathe it out. A specific carrier called CoA binds to the molecule as it comes through, forming a 2 carbon molecule called acetyl CoA. Like so. So our 3 carbon pyruvate was converted into acetyl CoA. Everything after this point, I want you to assume happens twice per molecule of glucose. So we have 2 link reactions, releasing 2 molecules of carbon dioxide, producing 2 NADH, requiring 2 CoAs. Acetyl CoA then enters the Krebs cycle. So I'm just going to do a little dotted line coming in, like so. It is involved in the transformation of a molecule which you don't need to know the name of, but you need to have heard because it might come up in an exam, called oxaloacetate. Hearing these names won't kill you, well, not unless you're very careless, but you don't have to know them. They will probably provide them for you if you needed them in an exam. The oxaloacetate is converted into another molecule called citrate, which is where the name citric acid comes from. Then another intermediary called isocitrate. Again, you don't need these terms, you don't have to have them memorized, just be vaguely familiar with them. It is then converted into a molecule called alpha, I like this name, alpha ketoglutarate. Alpha ketoglutarate, like so. In this conversion, another molecule of carbon dioxide is lost and leaves the mitochondria. Hooray, it's free. As well as this, the electron carrier NAD plus is converted to NADH. This moves to the inner membrane, where it is used in the electron transport chain. Let's zoom out so you can see that. There we go. It moves to the inner membrane, and I've just abbreviated electron transport chain. This is an high energy electron carrier, and that energy is going to be passed on to the electron transport chain to make copious amounts of ATP. Alpha ketoglutarate is transformed then into another molecule. I wonder if I've given myself enough room here. Succinyl CoA. And in this transformation, we get the same thing, a molecule of NAD positive being converted into NADH. And again, it goes to the electron transport chain in the membrane and is regenerated, like so. That's supposed to be an arrow, by the way. Okay. At this stage, carbon dioxide is also lost, another molecule. So if you're counting, we've lost 3 carbon dioxides for every 1 pyruvate molecule that we started with, effectively getting rid of those 3 carbons. Remember that, of course, because glycolysis produces 2 pyruvates from every 1 glucose molecule, that means that we've lost a total of 6 carbon dioxides, which we need to know if we're balancing our equation of respiration. Succinyl CoA is transformed into something called succinate. Again, these intermediary compounds you don't know the names of. And more, well no, this is not more. This is the first time this has happened. A molecule of ATP is produced from ADP and inorganic phosphate and an inorganic phosphate ion. So just a small amount of ATP. This is 1 per pyruvate, so that's 2 per glucose molecule. Succinate is then converted into another molecule called fumarate. Oh, these great names. Doesn't this make you want to be a biochemist? Succinate to fumarate, and this converts something that we have not come across before. FAD plus to FADH2. I'll just zoom in a little bit so that you can pick that up. This is another high energy electron carrier. It does exactly the same thing as NADH. It doesn't carry quite as much energy. It carries it over to the electron transport chain in the inner membrane, where again it is regenerated back into FAD+. It's transporting those high energy electrons to the electron transport chain. Well, they'll be used to make a large amount of ATP. Fumarate is then transferred into something called malate. And then back into oxaloacetate. This conversion of malate back into oxaloacetate actually produces yet another molecule of NADH. From NAD+. So this goes off again to the electron transport chain and is regenerated like so. Ta-da! So essentially, this is the whole of Krebs cycle. Very complicated. If you memorize the entire thing, but remember these names going around in the middle, you don't have to have. There are some things you do need to know though, and I'm just going to summarize that now. Glycolysis took place outside the mitochondria, producing 2 molecules of pyruvate. As it moved in via the link reaction, CoA was bound to the molecules, creating acetyl-CoA. In this process, carbon dioxide is lost and 1 molecule of NADH is made. Remember, this is per pyruvate. We then move into Krebs cycle, also known as the citric acid cycle, which goes around and around. Each time, 1 acetyl-CoA comes in, and various things are produced. Carbon dioxide, NADH, another molecule of NADH, carbon dioxide again, ATP, FADH2, and NADH. Every cycle, those are produced. Now, you may be asked about 1 glucose molecule or 1 pyruvate molecule, including the link reaction and the Krebs cycle, or they may just ask about the Krebs cycle if they're feeling particularly mean. So let's just talk about what our totals are in what is being produced. So, per pyruvate, and remember you need to double this if you're talking about, you need to double all of these numbers if you're talking about glucose. So out of 1 pyruvate, what are we getting? Well, in this, let's count the number of carbon dioxides. 1, 2, and 3. So I'm going to write 3 CO2 are produced per pyruvate. Let's see what else. NADH. We've got 1, 2, 3, 4 being produced. One is from the link reaction, 3 are from the cycle. And then, let's see, what else? Oh, ATP is also being made. We've got 1 molecule of ATP per pyruvate. And this other high energy electron carrier, FADH2. And we have just 1 of those per pyruvate. So double all of these numbers and you find out what we've got in for 1 molecule of glucose. Remember, it's very important to remember that the NADHs and the FADH2 are going to the electron transport chain on the inner mitochondrial membrane. I'll talk more about that in another video. ATP is going to go off to the cell to be used for energy. I'll draw an arrow out like so. And carbon dioxide is going to be expelled from the cell. So that's the Krebs cycle, the citric acid cycle. It's tough, but you need to know about it, so good luck.